Thanks, Andrew. And thank you, especially to David, for organizing this workshop. I'm really sad I couldn't make it in person, but I really enjoyed yesterday and to this morning's talks. So I'm going to be talking today about modeling phytoplankton trait adaptation. I want to start out by acknowledging my co-authors, my two postdocs, Nathan Walworth and Susanna Lillis, and my co-PIs, Sinead Collins and Martina Dublin. And let's see here. And this work actually came out of the 2017 trait workshops. Sinead and Martina and I spent many hours kind of hashing out and trying to think about how to move forward our models and our experiments to try to think about how phytoplankton trait might adapt. All right, so to start kind of a big picture, as many of you might know, my scientific focus really is at the large scale. What are the drivers of biogeochemical cycling in the oceans and how climate perturbations might impact the rates at which carbon, nitrogen, and phosphorus are cycled? So specifically, I'm interested in understanding how changes in the physical climate system, such as increasing CO2, global temperature increases, impact and interact with ecosystem dynamics and rates of carbon cycling. Specifically, we're focused on the micro scale. So we're thinking about the interactions between physical dynamics and marine microbes, focus on how different time scales of physical perturbations impact microbes, microbial community composition, and biogeochemical function. It's kind of the big picture where I'm coming from. But lately, we've been thinking a lot more about these questions in the context of evolution and adaptation. So given the short timescales of marine microbes, evolution becomes an important part of understanding what the future oceans will look like. And Ben gave such a fantastic talk this morning about kind of where we are with our thinking about putting adaptations into global models. But I agree with his kind of conclusions that we really need to understand where and how fast adaptation is going to occur in the ocean and what adaptive phenotypes are going to look like to understand carbon cycling dynamics and potential food web changes. So this talk today will be focusing on this kind of very big question of how phytoplankton will adapt to a warming ocean. So we've been seeing an increasing number of experimental evolution studies which show that phytoplankton can adapt on relatively short timescales, so 300 to 400 generations. And these experiments have now been conducted on a wide range of marine species, including coccolithophores, diatoms, trichodesmium, dinoflagellates, et cetera. And they've been done under both single stressor and so high CO2, for example, or high temperature, and multi-stressors, so combining multiple stressors together. And there have been many clear trends in how the trait values change as these populations evolve. We see increased growth rates, which we would expect due to selection, quantifiable changes in cell size, for example. However, in pretty much every study that I've seen, there's been a sentence or even more paragraph talking about complex phenotypic plasticity that were observed, or shifts in multiple metabolic, stoichiometric, and energetic traits. So what I see emerging is that even in these simple lab experiments with single studies, there are these complex phenotypic responses that occur. And so for me anyways, it's becoming more and more clear that in order to answer the question about how phytoplankton will adapt, we really need to think about how phenotypes are going to respond. And so that's a multi-trait phenotype beyond just single trait or even pairwise traits. And we need to do so in response to multiple stressors. So experimentally, this becomes intractable very, very quickly. So if you want to think about many different species responding to multiple stressors, I think Phil Boyd calls it like the nalgene problem. You just get exponentially increasing numbers of experiments that you have to do. So can models come to the rescue? And we had a really great discussion before lunch about models and modelers and experimentalists interacting. And I think we need to figure out ways to do this better and more frequently to have really true collaborations between modelers and experimentalists. And I think by doing that, we actually will be able to make significant headway on this challenge. But that's kind of a different talk. So today I'm going to be focused on where phytoplankton trait adaptation models are from my perspective and what I think we need to do to move forward. So again, Ben gave a really nice introduction this morning, so I'm not going to really talk about different types of phytoplankton models. This is just one schematic that I think was just kind of a nice illustration that oftentimes we have multiple phytoplankton groups, whether they are these kind of PFT functional types or trait-based types, and then nutrients and energy flows through these groups and through the trophic cascade. So one approach to modeling trait adaptation, one option, is to assume that the traits of our functional groups in the models evolve independently. And I actually had the slide in before Ben mentioned the Beckman study this morning, but I think this is a really fantastic example. I love this study of how single trait adaptation might occur in response to warming, so temperature optimal evolution in response to warming. However, for many traits, there are strong correlations between traits. And in fact, as modelers, we use these correlations oftentimes to define phytoplankton functional groups or functional types. So size is often used as a master trait. This is just one example of many fantastic data compilations that have occurred of cell size versus maximum nitrogen uptake rate for a variety of different marine phytoplankton. And you can see there's a really nice relationship between size and maximum nitrogen uptake rate. So a second option for modeling trait adaptation is to use these trait correlations and allow model organisms to move up and down along these trait-trait relationships. So for example, if we have our diatoms up here in the green circles, if they were to evolve to become smaller in the model, if we use this trait-trait relationship, they would start to look more and more like cyanobacteria. And sometimes we would expect these trait-trait relationships to be robust. We have metabolic scaling theory and other theoretical reasons why we would expect robust correlations between traits. But there are other times when these intra-specific relationships that we observe may not be good predictors about what individuals are going to do in response to an environmental stress. So this is, this is an example coming from Blasia Syra. This is part of our collaboration and coming out of Martina Doblin's lab, that if you look across a whole bunch of different strains, there is quite a nice relationship between cellular chlorophyll content on the x-axis here and cellular lipid content on the y-axis. So that's all the points. So this is our cross-taxa relationship. So if we were to put this into a model, we would predict that if chlorophyll went up, then lipid content would go up. However, what we found was that if you take these individual strains and you shift them into a stressful environment, so these blue dots here are in the kind of standard treatment, and you move them into either a high temperature, high light, or a low nutrient environment, the response is perpendicular to that across taxa response. And so the strains actually decrease their chlorophyll while increasing their lipid content. And this is because these are complex phenotypes and responding to these complex environmental stresses. But the takeaway here is that the model prediction using these trait-trait relationships would have been incorrect. It would have inaccurately predicted this response at the individual level. To make the story even more complex, there's strong evidence that the correlations between traits can and do evolve relatively quickly. So this is data from Sinead Collins using Chlamydomonas exposed to high CO2. And this is a summary table. So we're looking at pairwise traits, so trait A versus trait B, and then the correlation between those traits in the ancestral population in these yellow boxes, and then the evolved population in the red boxes. And the takeaway here is that if you look at trait 1 versus 2, again, what the traits are doesn't really matter for this point. There was no relationship between these traits in the ancestral population, but there was a significant relationship between the traits in the evolved population. And in other times, 1 versus 3, for example, there was a negative relationship in the ancestral and none in the evolved. So in order to model how phytoplankton will adapt, theoretically, we should try to consider multiple trait phenotype adaptation, so more than just pairwise traits. We have to include the fact that traits are correlated, that traits can evolve, and trait correlations can evolve. So an incredibly complex puzzle. To refine the question a little bit more, to get kind of towards more science questions, we started to ask, okay, what is the impact of ancestral trait correlations on determining the evolved phenotype? So if we have our ancestral diatom here and it has a bunch of traits, so that's our ancestral phenotype, and a bunch of correlations between those traits, so those are our ancestral trait correlations, how does these trait correlations impact this blue arrow, so the path of adaptation, and ultimately the evolved phenotype that we get? In addition, we'd like to know how reproducible trait adaptation is. If you run the same evolution experiment every time, many times, would you get the same answer? Are you always going to get this blue diatom, or maybe sometimes would you get the green one, right? And we know that in reality, you do get variability. And so how variable is it, or how variable are the possible trait combinations that come out? So as a really first step to trying to include these complex dynamics into models of phytoplankton trait adaptation, we developed this trace model, trait correlation evolution model, to try to investigate those two questions that I just posed. So I'm going to start off by presenting the concept of a trait-based fitness landscape, and to shorten that, I'm calling it a Traitscape. And I think it's a useful framework for thinking about how traits will adapt, but I'm going to walk through it so hopefully I don't lose anyone before I get to the actual model results. So the idea here is that for a population, you'll have a bunch of trait measurements, A, B, C, et cetera, and there's correlations or relationships between those traits. And we can synthesize this information efficiently into principal component space. And so you can create this PC space where each of these points are associated with trait values, a set of trait values that we've measured. But also this space captures the underlying correlations between traits that we see. Furthermore, the existence of strains within this PC space, so the actual data points that you measured in the lab, tells us where plausible high fitness phenotypes exist, because we know that these are populations that are realized. So we can kind of rotate this plot in 3 dimensions, and what we get is the X, Y axes are defining this PC space, which tells us about the relationship between traits, and then the Z axis tells us locations where we see actual phenotypes existing, or high fitness regions. And again, whether or not there's valleys here will in part depend on whether or not we have measurements in a given place. And I'm going to come back to this at the end of the talk. So this is called a rugged fitness landscape in the evolution literature. Again, I'm calling it a trait space because it's trait derived, and instead of having a long word, I'm just shortening it. And these traitscapes are environment specific, and they're going to provide us information about where high fitness phenotypes exist in for a specific environment. So once we have our traitscape, we can try to start to think about how populations will evolve or move across that trait space. So if we plop a population into a new environment, it's the ancestral population, it will be fairly maladapted. So it's going to have a low fitness for that environment. And over time, it's going to adapt to the new environment. So move across this landscape to higher ground. So it's going to try to find a fitness peak, as illustrated here. However, the direction that you travel is going to be dependent on the trait correlations of your ancestral population or your current population. In the direction that you travel is therefore going to also determine which of these peaks you end up on. And this behavior is consistent with other evolution models, but we're doing this in a kind of a trait space using this PC framework. So this framework would predict that the resulting phenotype that would evolve in a given environment is going to depend on where you start and your underlying genetic architecture or your trait correlations. Because if you start here in the black and you move this way, you're most likely to find this phenotype. Whereas if you move this way, you would be most likely to find this phenotype. Alternatively, if you shift your ancestral population to being over here at the pink dot, it would be very hard for you to find this phenotype way over here. And this is another aspect of rugged landscapes, is that it's easy to get stuck on high fitness peaks, even if it's not the absolute best phenotype in the system. Because if once you're up here on this high fitness peak of a certain phenotype, in order to move over here, you have to go through a fitness valley, a maladapted phenotype. And again, I'll come back to that with some cool experimental data at the end of the talk. So we can use actual data to construct these traitscapes. And so for this study, we use the Clemenomonas data I mentioned briefly. We can use them to construct these PC spaces. And we also know where ancestral populations occurred and where the evolved populations occurred, which is just a start and an endpoint for our model simulations. And then we can model an adaptive walk across this fitness landscape to try to understand how differences in that ancestral population in terms of the correlations, the diversity of correlations in the ancestral population, whether they're all identical or varied, and the start location impacts which phenotype or which peak we end up on. So we're using a modified Fisher's model for those of you who are familiar with evolution. So just briefly, we start with a bunch of individuals, 1,000 individuals. We have a bunch of traits. Each individual gets a set of traits and a set of correlations between the traits. Then at each time step, the majority of the population is going to experience a single trait change, and then the rest of the traits are updated according to the trait correlation. So this is just acclimation. And then a small fraction of the population is going to experience a mutation or a change in the trait correlation, and then the trait values will update. We can then estimate what the fitness of each individual is using our Traitscape, and applies selection where the most fit individuals are most likely to persist in the system. So to show you what this looks like in terms of the model results, the model population starts here in this yellow dot, and this is a single peak high fitness area. And so they're going to be moving towards this high fitness peak. And initially, these color here are the density of the population. So initially, we have a really diverse population with a whole bunch of different phenotypes that exist. And eventually, over time, as you would expect, they're going to converge on this high fitness peak, which we are selecting them to converge on. Okay, so they're moving pretty linearly from start point to end point if you look in PC space. But if we look in trait space, we see a nonlinear behavior, which is actually much more consistent with what we would see in these evolution experiments. So this is, the start population and end population, and you can see the whole population increases its fitness. It's moving uphill in the fitness landscape, which we expect. But if we look now in terms of I pulled out two different trait relationships. This is the start point, and this is the path that the population took to the end point. And you can see it's not moving linearly from start to end, but rather moving in a seemingly nonlinear path. And that's, again, due to this underlying structure or correlation between traits, which are also evolving over time. All right, so how reproducible is trait adaptation? Do we see multiple phenotypes emerge? And the answer is yes, we see multiple phenotypes observed, but not an infinite number of possibilities. So it's a stochastic model. We have to run it many times. And so that gives us an estimate of the kind of variability that one might expect in the system. And what we see is that the model repeatedly found 4 different solutions or 4 different phenotypes when we ran the model multiple times, but not each phenotype was similarly accessible. So this first phenotype, it found a bit over half the time, whereas these other 2 populations, it barely found at all. And then this 4th population found a kind of intermediate amount of time, the number of simulations. And if we switch where we start, switch that ancestral location, so switch from the black to the pink, we see that the accessibility of the phenotype shift. It still finds the same 4 phenotypes, but now it's primarily finding this middle phenotype. So the schematic that I showed is a fairly good representation of what's happening in the model in terms of the accessibility of the points being determined by your start location. But there's not an infinite number of phenotypes that emerge. So the second question we asked was, what is the impact of ancestral trait correlations in terms of determining the evolution? And so in the interest of time, I'm not going to show you a bunch of plots, but give you the takeaway, and I can talk to you more if you're interested in the details. But what we did here was ran a bunch of different tests where we randomly assigned correlations, where we assigned observed ancestral correlations for 1, 2, 3, et cetera, trait values. And what we found was that if you impose ancestral trait correlations on the initial population, the adaptation rate is slower, and it found fewer of these potential high fitness phenotypes. And conversely, as you might expect, if you impose evolved correlation so they can move efficiently through the PC landscape, the adaptation is much faster, and they were able to access more of these phenotypes. So the kind of takeaways that we took from this model simulation, which I know is very theoretical, is that these ancestral trait correlations really help determine accessible phenotypes and the rate of adaptation, and their reproducible phenotypes are emerging. And as I mentioned before, a lot of these results are very consistent with what's been seen in the kind of evolution literature. But the cool thing here is that we're able to take trait data, create this landscape, and actually model an observed experimental evolution study. And so we wanted to kind of test this idea a little bit more. And so Sinead asked, what happens if you bump a diatom off its peak, right? So we have our diatom sitting on high fitness peaks. Theoretically, we are predicting that there are valleys in between these peaks. So what happens if you bump a diatom off its peak? Is it going to go back to its same peak, or is it going to find another peak? So there are three potential outcomes here. So we can have an isolated peak where you bump it off, you create a maladapted phenotype, and then it will just rebound back to its same original phenotype. You can have connected peaks where you have a population, you bump it off, put into a valley, and it finds not its original phenotype, but a different one, but one that you've seen before. And then the third is that you bump it off its peak and it finds a new phenotype, which we haven't seen before in our culture collection or our data. And so Sinead and Jana made this Herculean effort where they took many different ancestors, many different replicates. They did a bottleneck experiment and then a back selection experiment. And so the teaser for this work, if Jana, she was here this morning, I don't know if she's here now, but if you see her, definitely ask her about it. The teaser is that we saw all three of these things happen in our diatom population. So we saw a lot of cases where isolated peaks where if you put them through a bottleneck, they returned back to the same phenotype that they started. But we also saw connected peaks where these phenotypes shifted to other phenotypes we'd observed, and some cases where they actually were able to find novel peaks or novel phenotypes. So I'm running short on time, so just kind of give you the takeaways. I would propose that there's a third option. It's more complicated, but I think it's more realistic. And I think we are starting to get to ways that we can actually do this, where we need to actually incorporate the impact of trait correlations and how that impacts the evolved multi-trait phenotype. And so we're working to create this eco-trace, which is incorporating these concepts into an ecosystem model. And so one step bigger, why this matters, why we should care. Well, I think it actually has the potential to be quite significant when we think about the future oceans, because if our traits can evolve and our phenotypes can evolve, that has implications for obviously export. If we think about cell size, export, if we think about changes in cell size, but if we think about other things such as carbon and nitrogen ratios, that obviously impacts biogeochemical cycling and export, but it also impacts grazing and higher trophic levels. If we change lipid content, that obviously impacts energy transfer across the food web. So if we want to get our whole marine ecosystem predictions correct and understand the sensitivity of the system, we need to be able to think about how these traits are adapting as we move forward. So with that, I thank my fantastic group at USC, but also this amazing collaboration funded by the Moore Foundation. Also, the NSF and Simons Foundation contributed to funding for this work. And I'm always looking for great postdocs to join the group. And with that, I'll take questions.
Questions?
Thanks. That was a great talk. I'm wondering if you could comment a little bit on how you think adaptation rates would vary based on whether you have connected peaks or isolated peaks. For example, if you're looking at the evolution of a thermal optimum, you might imagine more of a gradient system as opposed to a phytoplankton being advected into a different nutrient regime or something.
Yeah, that's a really great question. So I think kind of a two-part answer. So I think that the way we're thinking about it is really holistic. So there are cases like temperature optima where we're thinking about how the specific traits will work. Makes a lot of sense. But I think in general, it seems like we have probably more of this kind of rugged landscape than a continuum. But some of these peaks are quite broad. And actually that came out of the model, which was kind of cool. But right, so sometimes the phenotype is kind of diverse and sometimes it's more singular. And I guess the question is how many peaks are there and how deep the valleys are between them. And I think what we're seeing from the bottleneck experiment that Sinead did is that sometimes these peaks, the valleys are really deep. And so it's really hard to get across the valley to a new peak. And so that would be kind of your isolated peak example. And then sometimes they're not. And it looks like it's actually not so difficult to get out of the valley into another fitness peak. So I think the short answer is we don't know yet. But I think especially I would expect it to vary based on the kind of lifestyle of the different groups that we're looking at, right? Whether you are Prochlorococcus or Synechococcus, which is everywhere, or whether you're kind of a boom and bust type species, which are going through much larger population sizes changes.
Okay, we have a couple questions online. Jonas, you're first.
Hello. Thanks for a great talk. So I'm wondering, like, one thing that surprised me a little bit, but maybe this is true. So that for any given sort of trait combination, possibly trait correlation combination, we can sort of assign a fitness, and we get a fitness landscape in trait space, and then we can sort of project trait space using a PCA. But what I'm less sure of is, like, will the upward slope in the fitness landscape in trait space necessarily translate into an upward slope when you sort of project it using PCA? Will you sort of preserve the climbing once you're projecting down to a lower dimensional space? Or did I misunderstand something fundamentally about how you set this up?
Yeah, no, that is a really great question. So what I didn't go into, because it was a short talk and didn't want to get too complicated, is that in the PCA space, we actually end up with cryptic peaks. So within a single fitness peak in PC space, we actually, if you moved it to the kind of illustrative example I was showing in phenotype space, you would have multiple peaks. So they're overlapping peaks, and that's why we ended up with these multiple cryptic phenotypes converging in the model, even though we only gave it a single hyphenous location. And so that is a really great question. We haven't analyzed the slope, but the slope should be dependent on the correlations of the relationships of the different traits and how they're projecting onto the different PC axes. And so I would anticipate that there should be different steepnesses, and we definitely see different widths of the peaks, where some of the phenotypes end up having really broad peaks and some have very narrow peaks that emerge from this formulation. But it would be really cool to calculate what those gradients actually are, which we should be able to do.
All right, thanks.
Okay, so we have time for one more online. Kyle, you want to go?
Yeah. Thanks, Naomi, for an interesting talk. One thing I was wondering was in ecoevolutionary models, the fitness landscape is usually frequency dependent. And I was wondering if that is part of your model, or if you had thought about how that would like affect the dynamics you're looking at.
But can you clarify a bit in terms of?
Well, so like in an adaptive dynamics model, the like fitness versus trait relationship, the shape of that changes as the trait of the population changes. And that's how you get things like evolutionary branching, for example.
Yeah.
So I was just wondering how to think about that combining with what you're doing.
Yes. So I think that the short answer is that that this framework gives us different kind of widths of those selection peaks. So that it's in there, but it's done in a different way. And so it's not a tunable parameter or something that we're so the framework is fundamentally different because we're trying to relate it back to traits and how the phenotypes of the populations are evolving. But we are giving them these widths in terms of their mutations and their mutation rate and fraction of the populations that are getting mutations that are applying to the correlation values. And those widths are trait relationship specific and evolve over time in the population. It's probably easier if we talk and I can draw things out for you.
Sure. Yeah, no, it's a complicated problem. So thanks anyways.
Okay. Thank you, Naomi. Thank you, questioners. So let's thank Naomi again.